Viruses aren't just your everyday bugs. They are relentless, highly tuned biological machines. Think of them as master thieves. Their mission is singular and, honestly, a bit terrifying: to breach our cellular secure facilities, hijack the local machinery, and endlessly copy their own malicious genetic code. In this explainer, we're going to unravel this ultimate microscopic heist together. So, it begs the question: how exactly do microscopic viruses hack the human body? It's really the core of everything we're exploring today. How do these incredibly tiny, seemingly simple infectious agents pull off a hack of something as complex as us? I mean, they don't have brains, they don't even have their own energy source. Yet their infiltration strategies, honed over millions of years of evolution, are absolutely genius. Okay, let's dive into this. The primary objective for any of these biological hackers is viral replication. Simply put, they need to turn the host's cellular machinery into a dedicated factory that does absolutely nothing but mass-produce viral genetic material and proteins. It's a complete hostile takeover. But exactly how they manage to do this? Well, that depends entirely on the specific type of code they carry in their payload. And this brings us to the two main camps of our single-stranded RNA hackers: positive sense versus negative sense. Think of positive sense RNA as ready-to-read, plug-and-play code. It acts exactly like our own cellular messenger RNA. Our host ribosomes see it, read it, and immediately start printing out viral proteins. But negative sense RNA? That's backward code. The virus literally has to bring its own special molecular tools, specifically an RNA-dependent RNA polymerase, right into the cell just to reverse engineer its genome into a readable format before the heist can even begin. Act 1. The plug-and-play hackers. So with coronaviruses, because their genome is essentially ready-made mRNA, they walk right into the cytoplasm and boom, immediately their code gets translated. The virus hijacks our host ribosomes to translate its initial proteins. These process down into a polymerase protein complex, and then, get this, they literally remodeled the cell's interior. They form these double membrane vesicles right in the middle of everything, essentially building a secure safe house inside the host cytoplasm to hide their massive replication process from our cellular security systems. And here's one of their most efficient, honestly kind of crazy hacking tricks: discontinuous transcription. The virus doesn't just read straight through its genome from start to finish like a normal book. Instead, it literally jumps around to create subgenomic mRNAs. It intentionally skips parts of the track to rapidly print out the specific structural proteins, things like the spike and membrane proteins, that it desperately needs to quickly build its outer shell. If we break this complex maneuver down, it happens in 3 rapid-fire steps. First, it starts RNA synthesis. Second, the machinery actually jumps between specific markers, the transcription-regulating sequences. It leaps between the leader sequence at the start and the body sequences that precede each gene. And third, it recombines. This allows for incredibly high rates of recombination. It's fast, it's lethal, and it is plug-and-play coding at its absolute finest. Act 2. The reverse engineering hackers. Now, when we talk about reverse engineering hackers, we are largely talking about viruses like influenza A. And before we get too deep into their molecular mechanisms, we need to ground this science in reality. Just think back to the pure devastation of the 1918 influenza pandemic. It's a stark, really powerful reminder of what is actually at stake here. These microscopic hackers aren't just abstract concepts in a lab. Their ability to successfully hijack our cells has caused, and for sure continues to cause, massive global devastation.
So the crucial point is how influenza actually pulls this off. They have a segmented negative sense genome. So instead of one long string of code, their genome is chopped up into 8 distinct gene segments. And here is their sneakiest, most dangerous move by far. While most RNA viruses just hang out in the cytoplasm, influenza sneaks all the way into the host cell's most secure vault, the nucleus itself, to execute its replication. No way, right? But it does.
Once it's inside that nucleus, it performs an act of pure molecular espionage called cap snatching. Because its backward code needs to look like legitimate host mRNA to pass security, it just straight up steals the cellular ID badge. The virus literally snips off a tiny 10 to 13 nucleotide cap from the host's own messenger RNA and uses it as a primer to start synthesizing its own viral mRNA. It's brilliant and terrifying. What's really fascinating about this process is how the virus uses a specialized 3-part complex to get it done. Think of the PB2 subunit as the scout. It grabs the capped host mRNA. Then the PA subunit steps in, acting like molecular scissors, performing a precise cut to chop off that cap. Finally, PB1, which is the actual viral polymerase, uses that snatched cap to start reading the viral RNA. It is a flawlessly coordinated three-man inside job. But getting an ID badge at the front door isn't quite enough to completely fool the cell. Our human cells put a long tail of adenines, a poly-A tail on the very end of our mRNA transcripts. To counterfeit this final security feature, the influenza virus uses a wildly clever process called polymerase stuttering. The viral machinery literally slips backward one nucleotide over and over again on a specific track. It's essentially an intentional glitch in the matrix, allowing the virus to properly format its counterfeit code. In this continuous stuttering cycle, the polymerase arrives at the signal, inserts an adenine, moves forward, and then actually slips back one spot. It inserts another adenine and repeats this cycle. This repetitive slippage happens hundreds of times to perfectly construct that required poly-A tail before it finally releases the finished, fully counterfeited mRNA. Oh, and influenza has one more trick up its sleeve: leaky scanning. Sometimes the viral RNA features a really weak start signal. Because it's so faint, the host scanning ribosome essentially blows right past that first stop sign and initiates translation further down the line on a second start sequence. This hacker trick allows the virus to squeeze multiple, entirely different proteins out of a single, overlapping sequence of code. Talk about maximizing your payload, right?
Act 3. The double agent hackers.
Now for the biological rule breakers, like the arenavirus family. They use what's called an ambisense genome. Basically, this means they play both sides. Right in the middle of their RNA segment, there's this tightly folded hairpin structure. And at first, RNA synthesis just hits a wall and terminates right there at that hairpin block. But by playing both sides simultaneously, they manage to produce their required initial proteins early on. They synthesize from the 3' region of both segments, creating mRNAs from both the negative sense genomic strand and the positive sense complementary strand. And how do they finish the job? Well, they eventually manage to overcome that hairpin block entirely to synthesize the full-length segments and their final structural proteins, completely finishing the heist. Which brings us to a really critical question for all of us. Could understanding these exact microscopic hacking mechanisms, the cap snatching, the stuttering, the discontinuous transcription, be the key to stopping the next massive global pandemic? By knowing exactly how these invaders breach our cellular secure facilities, scientists can design highly targeted antivirals to jam their molecular scissors or break those stuttering cycles. This microscopic heist is happening inside of us right now, but understanding the hacker's playbook, that is our ultimate defense. Thanks for joining me on this explainer, and keep questioning the microscopic world around you.